It's my great pleasure introducing Dr. James Berg. He's visiting here at OIST from this month until February next year, if I remember correctly, as part of the TSVP fellow. He's here with two kids who are probably at school at the moment, but also his wife, Grace, in the audience. So I wanted maybe to start by giving a big round of applause to welcome the whole family to OIST. So welcome. So Jim trained as a neuroscientist. His PhD is in neuroscience from Harvard. And in 2012, he joined Allen Institute. The scientists are working with a famous scientist, Hongui Zheng, on neurophysiology projects. But since 2020, he's been the director of scientific operations in the new branch of Allen Institute called Allen Institute for Neurodynamics. So the Allen Institute is famous for having contributed a lot of super useful resources for the neuroscience community. Probably the most famous one being the mouse brain atlas, which probably some of us still use daily almost, along with other brilliant projects. And these big efforts don't just happen by chance, as we have come to appreciate quite acutely, having attended quite a lot of workshops and symposia trying to do some cohesive big projects together. So we are really incredibly happy that Jim's here to share with us today how to do team science well. So thank you so much for being here.
Okay, thank you. Can everybody hear me okay? Great. Yeah, so thank you, Azumi, for that introduction, and also for all of the work, the conversations and things kind of leading up to this opportunity. I really appreciate it. Also, thank you to the TSVP program, Jonas and Lynn and the entire TSVP team. It's been really, I'm just kind of beside myself with how just incredibly flexible they've been and welcoming and kind of to me and my family. Really like kind of it's above and beyond anything that I could have expected. So I think that this TSVP program is a model for how to kind of make collaborative science easy. I really appreciate it. Yeah, so I'm Jim Berg. My pronouns are he, him. And I'm Director of Scientific Operations for Allen Institute for Neurodynamics. This is actually a return trip to OIST for me. So in 2011, I was a postdoc at UCSF, and I attended a course here at the Seaside House called a course on molecular neuroanatomy. That course was put on with the Allen Institute for Brain Science in collaboration with OIST. And so I'm kind of way here in the back, right? That's me right there. So I had a really incredible 3 weeks there. So it was 13 years ago. It was right when it was right when the OIST was being was kind of launching, and so I was a tourist for parts of it and took some photos and saw kind of I think it's Lab 2 under construction up there and the auditorium, and so it was OIST has always had a special place in my scientific heart for kind of having this, hosting the symposium with the Allen Institute, because that's also where I met all of these Allen Institute scientists, where I was quickly able to kind of, I was working on my CV as I was working with them, and was able to be recruited to join a physiology effort there the following year. So in October of 2012, I was joined the Allen Institute. So as Azumi said, I joined the Allen Institute for Brain Science in 2012, and then recently joined the Allen Institute for Neurodynamics as it launched. I wanted to do just a little kind of differentiation between these two institutes because neurodynamics is so new. So the Allen Institute for Brain Science was what the original Allen Institute was founded as in just about 20 years ago in 2003. And really the focus of that institute is on kind of cataloging the diversity within the brain, the mouse and the human brain primarily. They share those resources through kind of web-based resources and different APIs for kind of data access. In neurodynamics, we launched in 2021. and are focused on kind of collaborating with our sister institute, the Allen Institute for Brain Science, in kind of looking at those neuron types and trying to understand how those types interact to influence kind of motor action, behavior, decision making. We have a kind of model for sharing data based on this cloud-based analysis platform that I'll talk a little bit more about later. And I think kind of the glue that pulls these two institutes together is that kind of our overarching kind of goal is to kind of understand how the brain works ultimately to be able to influence kind of therapies to improve health. And I'm going to talk a lot about kind of different neuron types within the mouse and human brain. There's going to be a lot of them, and sometimes it gets a little bit overwhelming for me to kind of be thinking about kind of these cell types and how they interact. So I'd like to use a model to kind of start talking about neuron types and why we should care about them. And for this model, I chose the cricket. And so we're not studying the cricket, but I like to use this model that you can imagine being a cricket in the evening, chowing down in some farmer's field, really enjoying yourself, hopping around, no problems in the world, but there is a problem, and that is bats. So there are some bats that love to eat crickets. So this cricket has evolved a mechanism to avoid bats. And it basically kind of executes this algorithm. When it detects a bat, if the bat is on the left, it needs to jump right. If it's on the right, it needs to jump left to get out of the bat's like sonar zone. And so how does this cricket do it? Well, crickets have ears on their legs where they detect sound. Those sound sensors are, for the most part, used to hear other crickets and their mating calls, but about quarter of them or so are actually specifically tuned for supersonic sound to detect bats. And so these sensors sense the signal. The signal travels in to another neuron called the ascending neuron that sends a signal up to the brain. It's bilaterally symmetric, and that happens in a region called the prothoracic ganglion. So this is bisymmetric, and so as you know, there's a problem. They need to be able to jump left or right, and so they have another specialized neuron, this beautiful neuron called the omega neuron, and these omega neurons basically synapse on the opposing side, and they decrease the activity of that other side. So in effect, what this means is when the bat approaches from the left side, it activates the left leg / ear first, and then by the time the sound gets to the other side, the omega neuron has dampened that activity and allowed for the left signal to kind of go up. And the way that signal goes up is via something called a burst of electrical activity, which I've kind of represented here as these kind of spikes. And so that's those spikes, that's a burst, it's like an unequivocal signal to the brain. It's not nuanced, it's really that siren that there's a bat coming from this direction, and that leads to another kind of cascade of other specialized neurons that lead to the motor action for the cricket to jump right. So this the point I want to kind of make here is that what we're talking about is kind of specialized neurons contacting each other in kind of specialized ways, and then acting, having physiology in a specific way to lead to this, and admittedly simple behavior. And so this obviously humans and rodents, we execute algorithms as well. Kind of everything we do is based upon kind of combinations of these algorithms. You execute an algorithm to kind of come and attend this talk today. My family executed an algorithm to move to Japan for 4 months. Those obviously are not as simple as a couple neurons detecting a bat, but you can kind of see how it might be based on kind of many more neurons interacting in different ways to lead to the behaviors that we all execute. I want to show you, this is the omega neuron. It's just a beautiful cell. So I think if this doesn't excite you, then you're going to have a long talk ahead of you. It's just, yeah. So let's actually look at a mouse example of a mouse, some mouse behavior. So I'm going to highlight a data set of visual behavior, two photon calcium imaging that we have, where we record from mice as they're behaving, and this is a mouse that's undergoing a visual stimulus. It sees a certain scene, and it runs, and then it sees that same scene. That scene kind of repeats, and then when the scene changes, the mouse is trained to expect a reward, that it engages in a lick, and then it gets a reward. And so this mouse, their mouse is trained under the algorithm that if the image that they see now is different from the one that their last image, then they lick and they get a reward. So it turns out that some mice, or a lot of mice most mice use this strategy, but there are some mice that use a different strategy, which is basically just like wait a while and lick every once in a while and get lucky sometimes and get a reward. And so this is an alternate strategy that is just based on kind of lick based on task timing. There's different things where there's penalties for licking at the wrong time that lead to delays, and we try to kind of get the mice to do what we're trying to the algorithm we're trying to study, but they do both, and so we decided to kind of study both and look at what happens in the neuron population while this is happening. And so the data set that we have is looking at using a window to look at the visual cortex of the mouse while it's engaged in this behavior, looking at various different neuron types using calcium imaging. So calcium flashes when these neurons are active, and kind of looking at that activity while the mouse is engaging in this behavior. And we see kind of an interesting thing is that based on the strategy that the mouse is taking, and you can tell which strategy they're using based on their activity when they're licking, the activity of these neurons is different. And so, the activity of these populations of neurons is different. And so in the excitatory neurons, the pyramidal neurons that we measure, when the neurons are kind of using a visual-based strategy, looking at the images and looking for a change, we see higher activity levels when the mouse gets a reward after a successful attempt than if they're just using kind of a timing approach. The other thing that we do is we use a distractor, where occasionally we would just not show an image and have the mouse have to wait for the task to kind of re-engage. And during those omissions, there's no there's kind of no difference between the two different strategies the mouse is taking. This is in contrast to a couple different other types of neurons. One type called the SST neuron that's labeled by a specific creatine that labels these SST neurons. And in this case, there's the population basically spikes or has activity whenever the visual stimulus is applied, and it's pretty much no different between the two different strategies. But when after a reward, if the mouse was using the timing strategy, there's slightly elevated activity compared to the mice doing the visual strategy. And then during these delays, you get this interesting divergence where the timing strategy, when a mouse is using timing strategy, those neurons kind of increase gradually over time, whereas the ones using the visual stimulus kind of decrease. And this is kind of in contrast to another set of neurons kind of within there, the VIP neurons, that they're kind of excited during the intertrial intervals, not during the stimulus. They get kind of depressed during the stimulus, and then during the delay, you see a big difference between the VIP neurons and the mice that are attending to the visual stimulus rather than relying on time. So all of this is to say that there's these neurons in the mouse cortex that seemingly are the same. They're labeled by the same kind of pre-line, but kind of based on kind of what the mouse is thinking, they're behaving differently. And so one question that we have is whether these neurons are the same neurons in the mice that are using the two different strategies, or if they're kind of two different neuron types. And so for that, we need to look at kind of the cell type diversity within the brain. And then to really look at the cell type diversity, we start with transcriptomics or gene expression, or transcriptomics. The reason why we use gene expression a lot of times is upstream from protein changes and different changes in the properties and shapes of the neurons. So it gives us a good handle kind of on an upstream property. We know that altered gene expression can lead to disease. And then really kind of one of the keys is that this is optimized tools where the properties of studying gene expression with single cell sequencing approaches allow for kind of high throughput approaches where we can really take that complexity of millions of neurons and are able to catalog it. And then the other thing is that we can, if we're studying gene expression, that can lead to us engineering new tools, mice and viruses, which the Allen Institute has a big program in doing, basically feeding back from the transcriptomics to be able to lead to these new tools that we use, some like the mice that I was just talking about before. Just a quick kind of primer in how we do this with transcriptomics is kind of isolate a number of different cells or nuclei. We perform unsupervised clustering based on the gene expression of each individual neuron to cluster them with cells with like gene expression profiles, and then organize them into clusters. We then kind of annotate them and kind of try to take those data and make sense of them in a hierarchical approach. What this actually looks like in practice. So this is just mouse visual cortex interneurons, because that's some of what I was talking about before with those VIP and SST cells. We can look at gene expression in a lot of different ways. So one way that we look at it is on a UMAP plot, which is basically kind of spatially arranging the neurons based on that sparse principal component analysis that we do on each of those, on each individual neuron. So each dot here is an individual cell, and it's spread out based on its gene expression data. The neurons are colored based on a clustering algorithm to type them into the different types, be it parvalbumin, VIP, SST, or LAMP5 or SNCG, the major classes of interneurons in the brain. And then this can be kind of a little bit complicated to look at. So then kind of translate this to each cluster being kind of a dot on a plot like this and some lines between them, because as you can see that there's some types are discrete, are a little bit more kind of separated. Like this is actually a group called chattel of SST cells that are completely discrete, and it's actually this yellow one right here, whereas other ones kind of have intermediate cells kind of in between. So it's kind of our compromise between kind of continuity versus discreteness of neuron types. So this is really helpful to know that, okay, when we're looking at that SST cell in that behavior experiment, there's a number of different types that those could be, or combinations of types. Same thing with VIP. But really, what we want to know is how those neurons are actually behaving and shaped and acting together. So this is, plots is, I think it's a requisite kind of Hall image in most talks, that we want to know kind of the shape and the physiology, the electrical activity of each individual neuron. The way that we do that is using what we call slice electrophysiology and Patch-seq on tissue. And so this is an approach. This is from the Allen Institute. This is just from protocols.io, but I thought it was a nice image showing kind of where you take the brain, dissect it, slice it into thin sections, and then record from individual neurons, kind of by arranging the slice on the side. And so this is the Patch-seq technique, where I'm kind of summarizing it all in this figure right here, but this is a paper that we put out kind of detailing really in excruciating detail kind of how we got to kind of our final protocol here. It's a technique that was introduced by multiple different academic labs, but then we took it and applied it to thousands and thousands of neurons, iterating on the method kind of little by little, and kind of, I think, came up with what we think is kind of the optimized approach. I'd be really thrilled to kind of, if anybody wants to meet and kind of talk about these and are using kind of this technique, I'm really happy to kind of walk through our technique. But at a high level, what we're doing is recording from individual neurons. As we record from them, we give a specific kind of input signal to them and measure their output to get that kind of input-output characteristics of the electrical activity of the cell. We note kind of where we recorded from that cell, and then after the recording, we kind of nestle up, grab the nucleus, and slowly extract the nucleus out of the cell. By doing it slowly, what we allow to happen is for the neuron to kind of recover on the back end, to reseal itself, so that it can keep the dye that we've injected into there during the recording, and I'll get to that in a second. But we pull that nucleus out, we put it into a tube, and then we actually send that through the exact same pipeline that all of our fact sorted data. So one of the things we try to do is when there's already something that's working, we try to figure out like how can we get on that train and kind of insert ourselves there. So that's so we're looking at data kind of when we compare it with the transcriptomics data, we're really looking at it kind of it's coming off the same using the same probes and everything on the back end. We fill the neurons with dye and then react that dye and image the neurons and then reconstruct them to be able to tell their shape. And this just gives kind of general, like, we worked hard to basically kind of maximize the efficiency here. Our technicians they're better physiologists than I ever was. They can get almost 90% of the cells are able to get their nuclei out. Really exquisite ability to get more morphologies. An interesting thing that we talk about in the paper that I can talk about on the side with anybody is that there's a trade-off between morphology and transcriptomics. We really have to you have to kind of you could vary your electrode size a little bit to get better transcriptomics, but worse morphology, or vice versa. And so it's it was a really fun project to kind of optimize these conditions. So what's the payoff? What do we actually get when we record these inhibitory neurons? Sorry, really quick, just to kind of clarify that this is a plot showing the gene expression from Patch-seq cells compared to fact-sorted cells. And so this is really exciting because this is like it's like 6,000 fact-sorted cells and like 4,000 electrophysiology Patch-seq cells. And so it's really exciting to kind of get to a point where we had kind of really equivalent matches between these two different populations, so we can feel really confident that the data that we're looking at really do represent those dots that I was showing you on the plot before. And so when we start looking at the physiology, we find that the physiology of each specific transcriptomic type that I described before, that is based on dissociated neurons. When we look at the physiology of those transcriptomic types, they have really incredibly stereotyped features between cells within in the same type. I think this is where my son would say "Yatta." It was something that when we really looked at the data was really kind of just impressive to see kind of how stereotyped those responses were. It's something that as we were, I think it's nice to kind of look at it and kind of you can tell kind of there's some fast spiking cells that people might know of. There's stuttering cells. There's lots of cells. There's like delayed firing cells. There's lots of words that we use to kind of describe these cells and kind of catalog them. But we wanted to, especially because we had 4,000 of them, we really wanted to kind of we almost wanted to take a transcriptomics type of approach and really look at the data with a little bit less bias. It's not completely unbiased, but a little bit less biased approach. And the way that we do this is that we take the raw data traces, we do the same exact stimulus on every single neuron. So we're able to basically take all of the stimuli, the responses from the stimuli, and we concatenate them into kind of different groups of responses. And then when we can look at them kind of across, in this case, thousands of neurons and then apply principal component analysis to each of the traces and pull out differences between those traces. And then in the same way that we do with transcriptomics, we're able to represent that on a UMAP plot. And when we get a UMAP plot, we get something that looks like this, really kind of similar to what we saw before with the transcriptomics, where the major classes are separated. And then what we can do is we can kind of we can look at what is the landscape of that UMAP plot, kind of what is being represented there in terms of electrophysiological properties. And we get things like this is the APVM, which is actually this representation right here. A lot of this represents kind of the fast spike, the half width of the shape of the spike. And so you can see kind of the parvalbumin fast spiking cells are kind of the fastest up here, and this is one factor that kind of has a gradient kind of across the UMAP. There's other factors like this normalized subthreshold. This is if anybody has ever studied the SAG phenomenon in electrophysiology data traces that reflect this IH component of the trace, where so some of these neurons have kind of more or less SAG, and kind of on with different properties. I can kind of show this in an example of 4 different SST cells right here, where you might see kind of this neuron right here, this neuron right here. They both kind of have this like stuttering, irregular kind of firing, and you might think that they're kind of similar. But then when you start pulling kind of all of the data together, you can see that they actually live. This is the Chottle cell that lives kind of down on the east coast of SST, the continent, compared to this kind of more irregularly spiking cell is the HIPSI that's kind of up north. And then, as I mentioned before, this SAG is represented kind of in this area down here. And so this CalB2 type of neuron exhibits a lot of this SAG feature. And so, the main, I think the main point of this is that these, I mean, to kind of say simply, like the transcriptomic types, they represent real differences in physiology and likely lead to kind of differences in functional types of these neurons. What about morphology? So we also, we don't have as much morphology data, so we're not able to do the same kind of, the same type of kind of principal component analysis with morphology data, but we can label the neurons based on their T-type, and you can see that kind of similar to electrophysiology, the morphology of these different T-types is remarkably consistent. There's a lot of different types that you might recognize in here, from kind of Martinati types to here. This is a specific type that labels these chandelier neurons that have these kind of dangling axons, and I think it's just kind of remarkable the diversity of the morphology types within all of these classes. So then we talk about kind of like the previous the mouse study that I was talking about, kind of VIP neurons and SST neurons, but there's a lot more to learn kind of by using these data and kind of connecting it to those behavior data. I want to mention before I kind of get onto the next part that all of these cells are online at knowledge.brainmap.org in their categories next to the transcriptomics data, and so there's an easy way to kind of access the data and look into kind of your favorite neuron type. In terms of next steps for kind of bringing it full circle with the behavior data, really this is kind of showing that we can use transcriptomics as a kind of like a Rosetta Stone, a linker between different experimental approaches. And so what they're doing kind of literally maybe not right now because it's Thanksgiving, but what they're doing almost right now in the lab is they're doing these same experiments, and then they're dissecting out the mouse brain afterwards, finding the same exact neurons that they recorded from, and then they're using a technique called MFISH, multiplex fluorescence in situ hybridization, which is a similar technique to was in the Allen Brain Atlas that Azumi mentioned at the beginning, to basically do multi-round gene expression analysis and get a transcriptomic profile for every neuron that was recorded. And so we think that this will be kind of a link. This multiplex fish is going to be a link between the physiology data, the behavior data, and kind of the morphoelectric data in I want to talk a little bit about human cell types as well. So we study mouse, but we also study human. And so the human pyramidal neurons, when we did a study where we're kind of looking at the histology of a section, of a cortical section in the mediotemporal gyrus in the human, and saw this kind of incredible diversity between layer 2 and layer 3 as you get kind of deeper into that column. This is something that in the mouse, we tend to be in like kind of layer 2, 3 tends to be a pretty homologous, dense structure of these cells. But in the human, there's an expansion that happens in those layers, and that expansion is also represented in kind of a diversity of neuron types in the deep layer. This is something that's been known for a while, and these deep neurons, there's a lot of study on kind of these ginormous, massive neurons in kind of deep layer 3 of the human. These neurons, they express this specific, or they're labeled with this heavy chain neurofilament protein antibody. There's been studies in kind of human and non-human primate that they project long distances on the ipsilateral side of the cortex. They're really these long-range neurons that kind of connect different brain regions, integrate different brain regions. They're basal dendrites in humans during childhood and learning in kind of early childhood. Their basal dendrites undergo this like incredible amount of branching and diversification. And then these specific neurons are found to be kind of specifically vulnerable in studies of Alzheimer's disease. So we wanted to apply kind of the same approach that we just applied for the mouse into the human. Start with transcriptomics, as we mentioned before. So this is kind of a similar, this is gene expression UMAP, so not an electrophysiology, gene expression, back to there, where you can see that there's, in the supragranular cortex of the human, there's about 5 different excitatory types. And so when you map on the gene expression of that neurofilament heavy chain that I mentioned that labels these really big neurons, they tend to be kind of at the bottom of, to me, it looks like a sock. So it's like the bottom of the sock is where you find all these neurons. So we decided we wanted to use Patch-seq as a way to approach kind of looking at the morphoelectric characteristics of these cells. Well, we already had the pipeline kind of ready for mouse through a lot of iteration kind of back, over and over. And what we did is we collaborated with local neurosurgeons who remove pieces of human brain from people suffering from either epilepsy or sometimes deep tumors. You have to remove a little piece of the brain to get to a deep tumor. And so we collected, we're in the operating room, collect the tissue, run it back to the institute. We do slicing, and it's the same people doing the mouse slicing or doing the human slicing to prepare the slices. We do a lot of pathology because we basically we want things to be kind of on the edges of the tissue. We don't want it to be kind of in any sort of pathological region to make sure that we're in a good place to represent as healthy as possible given the situation. This is, every once in a while you have like one of those experimental days where just the experimental gods are shining on you, and so this is we call it a representative sample in our paper, but it's definitely was on the far edge of like a really amazing day where this is somebody who was patching. This is a technician who's patching this. Started at like 2 o'clock in the afternoon and was there until well after midnight, patching from the same slice, recording kind of just down the road, neuron after neuron after neuron. And she came. Her name is Lindsay Ning to give her credit. She texted me kind of late that night and said like, I just had the slice of all slices, and so then after we did reconstructions and everything had just kind of like, it was like 10 out of 10. So this is, but you can see kind of the representation of the different kind of spiking patterns in these human cells. They're different kind of beautiful these morphologies, and then the gene expression that matches up with the same types from the transcriptomics profiles. But really, we want to get into kind of obviously multiple samples over multiple days, multiple patients, and really look at kind of the diversity within these types. And really, we see kind of what we really what was predicted from the histology, that the superficial layers really have kind of stereotyped, really similar kind of pyramidal neurons. But then once you get into the deep layer 3, you get these really wild, large labeled neurons that are in these CARM1P1 type. And they're right next to these kind of teeny tiny cell bodies in the cold 22A1 that send up kind of more of a spindly dendrite into kind of more upper layers. And so really this is really kind of impressive, the amount of kind of diversity that we were seeing in these deep layers, and really especially the local dendritic branching of these neurons compared to the upper neurons that have a much more kind of a tree with a tuft. These are really like explosions of local, more local dendrites, really kind of, at least by morphology, looking like they're kind of more integrating information kind of in these deeper layers together. Kind of summarizing the physiology, the physiology was very different kind of between these different types. And one thing, there's a lot of SAG, kind of come back to SAG. This IH also helps with integration with large dendritic arbors. And so we see a lot of SAG in these deep neurons. And then we also see this like high instantaneous firing frequency, which reflects these like bursts of action potentials. I think especially if they're transmitting over long distances within the brain, that they might use a burst to kind of enhance that long distance signal. And then just to kind of bring things back to these identifying whether they are indeed these neurons expressing the neurofilament heavy chain, we did FISH, fluorescence in situ hybridization experiments, to identify these different types in human tissue, and then co-immunolabeled them with a SM32 antibody, which labels this non-phosphorylated neurofilament heavy chain, and found that especially the CARM1P1 has a lot of labeled, mostly are labeled by this by the antibody, and then the FREM3, the deep neurons are labeled by it. And so FREM3 is I don't have time to go through it today, but FREM3 is this really interesting T-type that kind of spans kind of both of those, both the superficial and the deep types. And I think it's one of these kind of, we a lot of times paint these pretty pictures of very distinct kind of cell types that are discrete, but I think the FREM3 type is actually an example of a cell type that slowly changes without any big changes that can lead to clustering it differently, but so it ends up being kind of a big diverse group that again, I'd be happy to kind of talk a little bit more offline. Okay, so that was, I think I'm good on time, because that was kind of most of what, I was at Brain Science for a while, and now I'm in kind of the Allen Institute. I'm in the Allen Institute for Neurodynamics. Allen Institute for Neurodynamics launched in 2021. I joined in 2020, like really during the craziness of COVID, joined the Institute. And what I've been working with Karel Svoboda is the lead scientist for neurodynamics. And so he and I have been kind of working together since 2020 to build kind of an approach that steals some of what I really felt like worked from the Allen Institute for Brain Science, and then approaches things in a new way for areas where I thought we could diverge. And so the rough structure that we try to execute in neural dynamics is we're trying to set up these strong data platforms that a single data modality with a team that operates them based on their expertise, And I think critically, we try to pull all of the data together into a single data architecture. So we, instead of, this is something that was in brain science, each data modality really had their own way of doing things. And so it was really tough to, like, once we started doing Patch-seq and doing transcriptomics analysis, it was physically, it was easy to do, to move the neuron into a different tube to go, but the data was actually pretty challenging to merge. And so we decided to start kind of from the very beginning to have all of the data with a single metadata schema, a single format for kind of intake, QC, and analysis. The four main kind of strategies that we're taking is kind of multi-scale electrophysiology, mapping the brain-wide circuits, kind of doing an anatomy profile, doing behavior analysis, and then optical physiology, which I think if anybody was able to catch Kenta Hagehara's talk earlier this week, he talked a lot about optical physiology, so that's good. It gives me one fewer thing to have to walk through today. We build projects on top of these platforms, and the goal of these projects is to really be to really pull data from multiple modalities. And I think we're still early on, so I'm going to present a few different projects that are still taking it from kind of a single data modality, but they're like, you'll be able to see that they're kind of on the verge of pulling together other ones. And so I'm going to use just a couple examples to kind of walk through this. One is a study we do on kind of dynamic routing, and another is on this norepinephrine signaling. So one question that we want to address is how different information is, how information flows in the brain. And so this is something where if you're watching something with subtitles on, you're both getting visual information, kind of reading the subtitles. You're also getting auditory cues, listening to the movie that you're engaged with, and your brain has to kind of take both of those flows and combine them in some ways to be able to kind of integrate that into a perceptive kind of what's happening. The way that we are tackling this is using multi-scale or multiple insertions of electrodes and using electrophysiology. And so the reason why we do this is we're kind of at this point where these neuropixels probes, where we can record from hundreds or thousands of neurons simultaneously across multiple brain regions. So in terms of that, different brain regions that could be integrating this information. It works. There's people using them in a lot of different species, including humans. And then we are in a state where we are working on kind of the next version of the neuropixels probes that have integrated optical fibers to be able to combine optogenetics along with the electrophysiology. So this is a lot, really, the neural dynamics right now, we're kind of like, it's like being in a garage at points where we're really building a lot of things. And so one of the things that we built is this modular insertion system that is kind of like a dome that sits over the mouse as we're recording it. It can allow for up to 12 different electrode insertions into the mouse brain at a single time. The whole thing lifts up and down, has modules for cameras, for lights, optogenetics. It's really, as it says, modular. We have we take an approach where we have open hardware for the DIY crowd, that if you want to build this, you can build it. But we also collaborate with NewScale to license it to them to build it their own. So it's kind of this. We use these collaborations as a way to help with dissemination, and we know that not everybody wants to build one of these, so people might want to buy them. We also engineered a new kind of window rather than a cover slip to cover the mouse brain. This is a window that gets 3D printed, that has holes in it that allow for electrode insertion and kind of multiple electrode insertion at the same time. That's called a shield implant. That's published this year. We also, for a subset of cells, not every mouse, but for a subset of mice, we use an pre-experiment MRI to map the brain regions before we do the recordings. And this took a lot of we had to change the head post to a zirconium head post and do a lot of work there, but that's working pretty routinely right now as well to be able to reduce kind of targeting errors. And the last thing we're doing kind of correlates with that optoprobe that I talked about, where we're really trying to develop a cell type lookup table to be able to identify where the spikes are, what the type of neuron is that the spike is coming from. So with that approach, where we record from a mouse in multiple brain regions, and we give the mice an experiment where they are either rewarded for auditory cues or for visual cues, and we alternate those back and forth. So the mouse is very good at learning this. It needs to attend to auditory or attend to visual to be able to do that task switching. We recorded about 70,000 neurons so far from about 31 mice, and it's still a partial data set, but I think it shows some pretty interesting neurons. So I just like to kind of show some of the stuff that we can learn. So here's just kind of 3 example neurons that we're looking at. This is just looking at trials going down. So either the mouse is looking for an auditory cue or looking for a visual cue, back to auditory, back to visual. So this is just kind of a repeat. And we find kind of an auditory neuron here in the auditory cortex. It just responds anytime that there's an auditory sound. It doesn't respond when there's a visual stimulus. And so that's a pretty classic primary sensory neuron there. But here are 2 kind of other neurons that one is in the frontal cortex that is not spiking at all very much during, maybe a little bit during the visual stimulus, during the auditory zone. But then once it's a visual context, this neuron is baseline spiking kind of all of the time, and then especially when there's a visual stimulus is giving a kind of a burst of action potentials. And when we switch back to auditory, it goes quiet again, and then back to visual, it starts spiking again. And then here's kind of like an opposite type of a neuron, but this is a neuron striatum that's spiking during auditory, when it's auditory context, and then it actually gets quiet during the actual stimulus, and then kind of back, and then it's quiet during visual, and then kind of back there. So this is kind of when you're recording from 50,000 neurons, you can really start to pick out some interesting cells that are behaving based on kind of integrated information based on context. We can do a kind of a summary of that by flattening out the cortex. There's lots of gray areas that we're still looking to, but here's kind of the striatum with a higher fraction of those context specific, and then really the frontal cortex has kind of the highest right now, the highest kind of fraction of these cells. But as we kind of continue to do more and more experiments, we'll be able to fill this out. Yeah. It's the cue is that so when it switches, in this case, the mouse knows that given an auditory cue that it needs to lick to get the reward. If there's a visual cue, it doesn't get a reward via a lick, and then it's given there's a signal that we're switching to visual attention. And then in this case, now it's the opposite, where it does not get a reward for the auditory, but it does for the visual.
So if you ask the mouse to detect the condition change based on the error, so how would that recognition of the new condition is processed in the brain?
That's a good question. So you're asking if how the mouse, you're asking about the kind of looking at these change areas.
Yeah, so without any explicit cue, you suddenly change the condition.
Yes.
You have to figure out that something wrong, and you have to change my behavior.
That is a good question. I think it's I'm not sure if we've tested that specifically yet. As you can imagine, it's always a balance between trying to get the mice to do what you want them to do and measuring something. But it's possible that they've tried basically doing that, in later rounds once they kind of get the method. Yeah. Okay. We're running out of time, so I just want to quickly, because this is like a really exciting new area too that also could potentially get to some kind of future collaboration. So I'd like to talk a little bit about this norepinephrine signaling, and I'll go quickly through it. But basically, in neural dynamics, one of the things that we're doing is trying to identify brain regions that have small numbers of cells that might impact a large number of, or that might have like oversized impact on behavior. And so the locus coeruleus is a part of the brain that in humans we know that its activity or in mice it causes anxiety when it's overactive. It can cause mice to wake up from sleep, and importantly, it projects really all over the brain. This is both in a human and then also in the mouse. The previous studies showed kind of, it's a small region here, but their processes go kind of all over the brain. And so what we wanted to know is whether kind of this is one consistent population of cells that project all over, or are they different populations that project to different brain regions. So we want to know, what do single neurons look like? The problem is that neurons, they're both small and big. They have these really small dendrites that you really want to characterize, but then they have long axons that might go over centimeters. So it's really difficult to study these kind of small and large things at the same time, especially with the axons. If you're doing a method where one of those axons images, if you have a tile and it gets broken a little bit, you could lose kind of the entire rest of the axon. So Richard Feynman has he really loved kind of biology, where he was like, in biology, just look at the thing, just make a bigger microscope and look at the thing. And so we just decided to take that approach. And so we have a pair of scientists that work on kind of chemistry of the brain histology and then imaging. We first, in order to kind of make for kind of to be able to trace these neurons, we first used this tissue expansion technique that I think it's done in kind of a number of labs. But basically, if you're coming up on the diffraction limited imaging, one way to kind of beat the diffraction game is to actually just make your sample bigger. And if it expands isotropically, which it does, you can kind of get increased resolution by making your sample bigger. It brings up a problem, though, is that now you have this massive brain to image. And with traditional microscopes, if you try to image this brain, it's going to take like a week or a month. It's really, really big. It's going to have a lot of those tiles. Be really difficult to match them up. And so what Adam Glazer, a scientist in our group, realized is that there's all these people looking at microchip fabrication. They've spent millions and millions of dollars in R&D to generate these ginormous objectives to do microchip inspection. And so by taking that ginormous objective and putting it into a traditional kind of modified SPIM confocal microscope, we're able to image the brain in far fewer sections. In this case, kind of 15 tiles that we make up. The data that we get using kind of sparse labeling of neurons, we're able to see kind of individual neurons. You can see their kind of individual traces that are both long range and local. Once we have the image with all those beautiful neurons in it, we have a team that is actively working on reconstructing them. And so this is in collaboration with Google. So luckily the Google building is like right across the street from the Allen Institute, and they have a lot of experience looking at satellite images, mapping out roads and other features from the satellite data. So we've basically taken that team, collaborated with them to be able to automatically trace the neurons that we image in this approach. By far right now, the biggest bottleneck that we have is kind of proofreading those. So it does a great job of getting kind of the basics there, but it's still about 6 hours per neuron to be able to reconstruct it. To be able to kind of proofread it to make sure that the representation is accurate. The good thing is that we're kind of recording those corrections to be able to feed back to the algorithm that does the automatic tracing. And so we think over time, that 6 hours, we're trying to get that down to 1 hour. But where we're really looking for collaborators is that if you have an interest in a specific type of neuron from a specific region, we can collaborate. We can give you the data that show that where you have those cell bodies where you're interested, and then if it's kind of a trade where we'll give you the data and you provide the proofreading, and then kind of everybody wins. We get the scientific question answered, and as well as contributing to the common data set. Very quickly to just show what this means, kind of how to answer a scientific question here. Now we're labeling those locus coeruleus neuroepinephrine neurons based on the color of where they actually project. And so the projections are actually modular, where different neurons project to the cortex, to the olfactory bulb. Some of them project to the hippocampus, and then others project to the midbrain, to the cerebellum, to the spinal cord. And so this is by using this technique where we're able to look at each individual neuron by itself, we can really kind of delineate these different types. When we look at it like this, we see the most interesting that some of them project just ipsilaterally, some of them project bilaterally based on the different type that they have. So there's even as it's kind of a tiny brain region, they all express, they all have neuroepinephrine. There's a lot of diversity there that you could really look at in a modular way. And now the next step is that we're adding in MFISH with this as well, so that MFISH is kind of going to be transcriptomic type will continue to be the linker, where then we can look at these neurons with the long projections, how they're connected with the morphoelectric types, how they're connected with behavior types. And the trick for that is that the expansions, all of that takes a really long time, and the RNA degrades over that time. So we're looking at different ways to preserve and anchor the RNA during the expansion process. I mentioned this kind of collaboration. We've had a lot of collaborations already. If you're interested in doing this, let me know and we can collaborate. You can also send us brains. If you have a brain sparsely labeled, we'll take care of all of the downstream stuff and send you the data in Horta Cloud 2 months later. And so that's another way that we can kind of collaborate. Jairam wanted me to mention we're so close. We're going to have a morphology portal where you can both see the cells that are eligible for reconstructing for proofreading, as well as the data that have been proofread. So it's like a one-stop shop where you can look and see, oh, this is a place where I can jump in, or this is a place where where there's a lot of data already. I'm going to skip through all of this to at least mention that the applications for our Friday Harbor course to be able to use this neural dynamics data and all of the cloud-based approaches that we have been looking at. Applications are opening up next week, and so look forward to those. This is a really great program. Another kind of advertisement. I'm here until mid-February. I know I went through a lot of stuff and I didn't leave time, but I'm here until mid-February. I'm happy to have coffee. I'm happy to talk at lab meetings, different things like that. So. Kind of more formally, working on kind of a mini course on using the Allen resources, mainly focused on brain science stuff, probably in December, January, still working out those details. And then working on, essentially with Saskia de Vries, a colleague of mine from Neural Dynamics, coming in late January, early February, and talking more about kind of neural dynamics data. So I'm here for a while, and really eager to talk about that. Also eager to talk about just like the structure of science. I know OIST and Allen Institute have a lot of similarities and a lot of differences, and so I think it's going to be exciting to compare and contrast. Lastly, I just want to especially, there's a lot of different people that went into all the work that I described, especially when I mentioned Honghui and Christoph, who were part of my team, really excellent mentors in brain science. And then Karel and I are just really good partners in kind of taking the science vision and putting that together into a program to be able to work together and answer these questions. So I think I'm over, but I'll stay as long as I can to answer questions. Thanks.
much for this fantastic talk. It just feels like we're about to solve the brain. So any questions from the audience? Oh, Kenji, please. Are there instruments?
Yeah.
Thank you. So I have many questions, but first of all, so for as a team science, for the Patch-seq of 4,000 neurons,
Yes.
how many people over how many years worked on?
Yeah, that's a great question. So I mean, it's tough how you define a person, because it's a group that does the slicing. It's a different group that comes in and does the recording. So we have our recording people that do the patch clamp recordings. It's like, it was like my dream. You get to show up at like 9 o'clock in the morning, and there's like slices ready to patch. And so you know, it's a group of 4 or 5 people that do the slicing, but they also do all of the dissociation for all of the transcriptomic studies. So it's a kind of specialized group that's really good at that. The patchers are probably like 4 or 5 patchers. That data set was probably over about 18 months, I would say, is probably about a year and a half of kind of core day in and day out. I need to do the math, you know, 6 or 7 recordings per day. If you go to the paper, we were able to shrink down the EFIS acquisition from like 30 minutes, which we did in our first paper. Our second paper, each one was about 2.5 minutes. So we really increased the throughput to be able to run through these and try to approach the transcriptomics through them. So that's that, and then we get the different team doing the staining, a different team doing the imaging, a different team doing reconstruction. So it's a couple dozen people kind of total doing.
Yeah, thank you. So another question is with this gene expression information and the electrophysiological properties, how is it feasible or not to construct like a Hodgkin-Huxley type model in a data-driven way?
So hard. I think so. A couple of things. I mean, so we do find. Let's start with the easy stuff. We do find a lot of genes that we expect to find in the neurons that have really narrow action potentials. We find this Kv3.1, and we know as a strong delayed rectifier potassium current that slams down the action potential. We find high expression of that. Same with like an A-type current with neurons that have these like delayed firing. It's due to this kind of A-type current. We see that the potassium channels we expect, and so they're there. But starting from, but there's enough, you know, variability, and this is, you know, like Eve Marder's work, and kind of you can there's like, you know, 4 different ways to combine different ion channels to get kind of a similar physiological property. I think we definitely see that there's just a beyond kind of the big markers. There's a lot of variability. We see a lot more. Interestingly, we see more in the morphology with different cell adhesion molecules and things like that we're able to correlate a little bit more closely with some of the morphology features, which is something where I would have maybe predicted the opposite, that morphology, I thought you kind of express a bunch of genes and then let it go, and physiology you want kind of more active, but it seems to be, at least in terms of our resolution, that's what we have now. But then we're also pushing forward with protein, different ways to stain proteins, label proteins, and so that's also going to hopefully get us closer to kind of building those models. Yeah.
Another question. So this dynamic routing, I'm also very much interested in this program. And then Zhaojin Wan several years ago kind of proposed the possibility of different types of inhibitory neurons working for the gates for the inputs and the gates for the outputs. So have you already found out the different properties of different inhibitory neurons in this routing experiment?
No, not yet. I think that's another using the windows to, especially if we can localize, you know, it's unfortunately the most context dependent seem to be kind of in the least accessible from a window kind of perspective, in that kind of frontal brain region that kind of curves over. And so I think not yet, I guess, is maybe the short answer.
To hear more.
Yeah.
Thank you.
Yeah, of course.
Thank you for the talk. Fantastic data set. My question is on more like a team science aspect of the of your work. For the first part of the talk, transdomics was sort of, as you said, was the glue to link different data sets together. So I was wondering, at the AIND, what might be the glue for the different projects within the institute?
Yeah, that's a great question. So what we think the glue is I didn't show it here, but we have different types of projects. Okay, so it's in this model. I didn't label it here, but we have different types of projects we call resource projects, discovery projects, and exploratory projects. These resource projects are kind of the ones that are closest to the platforms, and those are the ones that are like more like the surveys of large data sets of morphology, or large data sets of even dynamic routing is almost kind of getting into that foundational like resource, where it's something where you can plan it out a year ahead of time and say, we're going to need X number of neurons across Y number of brain regions, and that's what we're going to put together. And then those are able to be pulled together for the discovery projects that kind of build on top of them and take something that they learned from those resource projects and move and kind of build on to the next thing. So I think kind of having this balance between those two, because the discovery projects like this norepinephrine signaling, that's something where we just don't know what we're going to do 2 months from now. We have an idea what we're going to do 1 month, but not 2 months. And so for those, rather than say X number of neurons over Y number of brain regions, for those we say, do what you want within the bounds of what we have for 6 months, and then come back and tell us what happened. And so it's more like a time-based thing. And so it's kind of, we don't want to balance all the way towards one or balance all the way towards the other. But who knows? Maybe actually, as the institute grows, we will actually do more of the time-based things and fewer the resource projects if we really feel like the resources, there's like an endless number of neurons. It's almost like pruning. Like it's difficult to decide what not to do rather than what to do, because there's always these distracting, like, oh, this is interesting. Like when I showed in those neurons, there was one crazy neurons that's recording that's going to multiple brain regions. So it's like, oh, what is that guy doing? So anyway, so that's it's a challenge for us in project management to keep scientists focused on kind of what they said they were going to study in the first place and not get distracted. Yeah.
Just one more question. So you said task switching is one of the sort of a big question in the team. But so what behavior is your team interested in general, or that even still undecided?
Yeah. So we have this. The task switching with the visual cues that change and auditory cues that change. But I would say most of the work that we're doing moving forward is in different foraging approaches. So this dynamic foraging with this kind of 2-arm bandit task, where the mouse has to lick left or lick right with various changes between those two. That's a big one that the norepinephrine story is gonna is using right now. And then we have another group that and so that's Jeremiah Cohen is leading that group, and then Cindy Poo joined, and she's working on this behavior called looking at patch foraging, which is kind of virtual reality, where the mouse is running through these like virtual patches and has odors to represent when it enters one patch versus a different one, that the mouse can basically exhaust the resources within those patches or can decide to move to a different patch. It's this like amazing, I can't wait for this to kind of get more out of this. This is amazing, like kind of 2-dimensional patch foraging kind of exercise, where the mice are able to, within a day, are able to just adapt right to it, figure it out, and execute there. So we have them, and then we just recruited Carl Schoonover to lead a freely moving mouse behavior paradigm with different foraging with mice that are freely moving with different trained on different odors as well, because we want to do as much as we can with head-fixed mice, but then we also realize that there's many more inputs to kind of come in. The mouse is able to really move, and the NeuroPixels probes are now coming on board, where you can record from them as well. Yeah.
Thank you for the nice talk. I was curious about one specific thing. I mean, as you shown there that the different brain areas, how they are connected. And I was interested that from one specific brain area, another specific brain area, the projection neuron paths, is there some kind of database that we can understand in 3D that how the path comes in order to make slice so that the projection neurons remain intact and we don't cut them?
Yes.
Database. I mean,
Yeah. So I think so we have a data set called the mouse connectivity atlas that has like bulk injections of viruses in different brain regions and kind of getting multiple neurons. So that norepinephrine example, or an example where that is kind of going all over the place. And so we have those tracks are in the they get labeled down into what we call the common coordinate framework as a common mouse or reference atlas. And so I know that you can image those in we have an approach called a tool called the Brain Explorer, where you can look at those projections. I'm not sure about doing kind of virtual slicing in that tool, but the data are there. And so I think maybe we could talk a little bit more. I think with various APIs, you could probably pull that data set and analyze it, basically kind of look at kind of virtual slices through to see, to get kind of an optimal slicing angle. Good question.
Great. Maybe in the interest of time, perhaps if anyone has more questions, perhaps after this talk, you can come to the front. Or, he's going to be here until February, so there's plenty of time. Please email him if you want to chat with him. So, well, thank you very much.
Yeah, thank you.